So, dear students, today we will talk about method and technology in modern pathology, and we divide all this stuff on two groups. This on methods and diagnostic methods and diagnostic technology. Methods is more substantial and divide on biopsy, macroscopic examination of operational material or anatomical grossing, autopsy, and experiment on animals. It's mainly nowadays experiment on animals. It's used in scientific branch of pathology. But some toxins, bacterial toxins, and some artificial or industrial toxins make some probe on animals. Mainly these methods used in 20th century. But now we use animal experiment mainly in science. Technology, it's related to some technique of studying material, and we have such technique like microscopic study. Most widespread is light microscopy, modern method, digital pathology, different type of electronic microscopy, radiological method, molecular genetic methods. This is mainly not routine, exclude macroscopic and light microscopy, but it still exists in big labs, in diagnostic centers, and in huge hospitals. I give example, scientific experiments. It's some plan for experiments. It's an example. We talk about Helicobacter pylori, which we can first study strains, and later we infect some experimental mice or other animals, and make some study by histology, immunohistochemistry, and etc. and other methods. Most famous method related with pathology is autopsy. Autopsy is most ancient method. It means see by own eyes. It's usually it's macroscopic examination after dissection body cadaver with some purpose of identifying disease and define the direct cause of death. It's have very huge history from Roman Empire. And some famous anatomists make autopsy, discovering a new organ, new pathology. But in the medieval ages, this method was forbidden because it's due to some religion causes, and its rise of autopsy was in Renaissance epoch and New Age from 16th, 17th century. But in the second part of 20th century, amount of autopsies significantly decreased due lack of interest of clinicians, development of new instrumental methods like computer tomography, ultrasound, magnetic resonance. I love clinicians to know more about human body in significant case of its autopsy is not necessary. Also, autopsy produce some legislative problem because it's due some conflicts, due some last willing or willing of relations and the family. It's not easy to explain this very very complicated area of pathology. But an example, in Ukraine we have special law which regulated this activity, and this law starts from 2003 or 05, and in most cases family which against autopsy can forbidden this autopsy and take body for funeral without autopsy. But usually we talk on previous lecture. We have some causes for autopsy when people receive treatment in hospitals and dying from some after surgery or some in short period until one day, etc. Next reason for decreasing autopsy is cost of this procedure. Is cost, time of pathologist, time of lab technicians, and the chemicals, and some procedure for freezing body, and some waste, loss of water, electricity, etc. It's costly for hospitals, and nowadays don't give so many information, and usually hospitals trying to make some economy for this procedure, and this reason for decreasing these old methods. If we ask people on street about pathology, it's all people think it's some lots of autopsies. This point of view cultivated by different serials, films, or mass culture. But we work. Our main work is biopsies, and we will talk on next lecture about it. This example, a microscopic study on autopsy. We can liver with cirrhosis and hypertrophy of heart. And also we use macroscopic methods on grossing when hospitals, clinics send material to us, and we some individual organs or tissue. We describe this tissue and make some measurements and make protocol about all information. Often it's very valuable information for diagnosis and help us in microscopic diagnosis. Our main method is histology. This routine procedure based on staining tissues by hematoxylin and eosin, and we prepare microslides by using microtome make a very thin section. It's about 4 micrometers, 7 micrometers, and stain in a histotissue processor in different liquids, and starting from xylene, put in different ethanols for dehydration this tissue, and put on last stage in paraffin wax, paraffin blocks. And these paraffin blocks we use for cutting these blocks on microtome and prepare microslides. Is after staining, of course. This is auto stainer. It's homemade in Sumy State University. There's some robotic arm which put these cassettes with slides, non-stained slides in different liquids. It's opposite process. We start from more concentrated liquids, and it's hydration. It's process against which we do before the return water in slide, and staining in hematoxylin and eosine. Interesting is this routine staining. It's common for all countries around the world, and all countries use these stains, this hematoxylin. An example of hematoxylin prepared from some trees in South America, and and eosine, this is prepared from insects, also from South America. But this very stable staining, and probably nowadays these stains produced by chemical way, artificially. This paraffin filling station is device used for preparing paraffin blocks. Okay, this immunohistochemistry methods use on reaction between antibody and antigen. Antibody against different markers, which these markers located in nucleus of cell or cell membrane, or membrane and cytoplasm. It's combined localization. Usually, primary antibody against something pathological protein or some receptors, etc., marked by some colored product like diaminobenzidine or phosphatase, or another substance like particles of gold, or some fluorescein substance. After reaction and fixation this antibody, we washing tissue, washing this sample. In case when the reaction not done, all colored product is washed away. But in case of presence some marker, this is products located in very precise place on these markers or this substance, and make some secondary reaction and visualization of this reaction, and can see presence some biomarkers. As another case, we use microscopes with polarization light. On left slide we can see this amyloid in prostate, it's corpora annulata staining, but Conho-red stain. But it is for exclusion some mistakes. Additionally, we check this stain in polarized light with specific green light, green apple reaction, and this phenomena of mild cross, this like that, or that. And this verify presence of amyloid. It is the same with another substances. In some rare cases, probably with most for science use, electronic microscopy. An example, transition microscopy, electronic microscopy. It's in this case we can see some vesicles, calcifying vesicles in atherosclerotic plaque, or produce connective tissue by fibroblast and some changes in thyroid gland. Before, in 20th centuries, some rare disease diagnosed by electronic microscopy. It's an example is Cartagener syndrome, which tissues, specific tissues in bronchial tree lost glycocalyx and this villi, and this proof by these methods. And nowadays it's not routine methods. Also, we talk about scanning electronic microscopy. In this case, we can calcificate in heart valve and some surface with fibrils in bone tissue. Another method is fluorescent microscopy, fluorescent in situ hybridization. In case of if some genus have amplified, it's increased copy of this genus, we use this method for verification of this increase in genetic copy. For this purpose, in labs use histotissue and this like primer for against gene. Usually in practice, this method used for heteronew mutation in breast cancer for verification level of expression of this gene, and use in future therapy some target preparate. Next method is polymerase chain reaction. Is artificially increase amount of copy DNA which we trying to find. In this, in case it's coincidence, this primer in gene, he receive lots of copy of this gene, which very easy to detect in some sample. Nowadays, it's automatic devices and program used for this next generation sequencing. It's in this case in lab, it's possible to make some copy several genes and find this mutation in huge amount of genetic information. This process cyclic and automated, and make this work in thousands times faster in comparison from 20 years ago. An example, it's nowadays it's a last word of science in this diagnosis. Usually this method used for genetic mutation in different tumors, this lungs, breast cancer, and some screening of genetic mutation. Very useful method. Also, we live in age of digitalization, and in all people hear about ChatGPT bot and this artificial intelligence, and probably in short time this artificial intelligence help us to make diagnosis. An example of a department with computer science department of our university, we also work on project for automated diagnostic program for breast cancer and prostate gland. And recently, 1 month ago, we made publication in the journal Pathology Informatics. It's Q1. And the journal, scientific journal, and the official journal of European Society of Digital Pathology. But this journal located in Indian origin. But last year this journal was this Elsevier by this journal and make official journal in branch digital pathology. Okay, thank you so much for attention.